This is lecture 5, part 2. We talked in part 1 about respiration, cellular respiration, involving glycolysis, pyruvate oxidation, Krebs cycle, and electron transport chain, which also includes chemiosmosis, and of course that's oxidative phosphorylation. And now we're going to talk about fermentation. Now, in aerobic respiration, the final electron acceptor in the electron transport chain is molecular oxygen. During anaerobic respiration, the final electron acceptor is not oxygen. The overall total energy yield is much less in anaerobic than aerobic respiration. And only part of the Krebs cycle and some electron carriers can operate under anaerobic conditions. Basically, they're not operating in generation of energy, and what you're getting instead is other molecules could act as electron acceptors, but we're not going to get into that. What the main thrust of cellular metabolism, this goes for many bacteria and for many eukaryotic organisms, is that you get fermentation. Now, during that fermentation, you do have glycolysis operating, so you get oxidation of glucose leading to pyruvic acid, and you get generation of ATP in this way. The pyruvic acid can then undergo fermentation, and there are two types of fermentation. One is lactic acid fermentation, and the other is alcohol fermentation. Some organisms can do lactic acid fermentation, others can do alcohol fermentation, and some organisms cannot carry this out at all. In our cells, we don't do alcohol fermentation, we do lactic acid fermentation. We do produce lactic acid or lactate. We do not produce alcohol. But there are many organisms that can, microbial organisms, bacteria, some yeasts, that can carry out alcohol fermentation. So fermentation involves release of energy by oxidation of organic molecules like carbohydrates, glucose, for example. It does not require oxygen, but can occur in its presence. Even though it doesn't involve oxygen at all, it could occur in its presence. Certain organisms can only carry out fermentation. They cannot carry out aerobic respiration. It doesn't use the Krebs cycle or the electron transport chain. Final electron acceptor is going to be an organic molecule. When we talk about final electron acceptor, we mean that whatever energy is left will go into an organic molecule, right? Only small amounts relative to aerobic respiration, only small amounts of ATP are produced in this process. So again, here we see we're looking at fermentation. This involves glycolysis, ATP production, and production of some high energy products by fermentation. It's the fermentation end products that are taking up electrons, and for the most part they still have quite a bit of energy. After all, I think it's fairly easy to understand that if you have alcohol fermentation going on, for example, one of the end products being, in terms of carbohydrate skeleton products, being alcohol, that has a lot of energy in it. All you have to do is put a match to it and you'll know that. So glycolysis, as you know, is the oxidation of glucose to pyruvic acid. Glucose is a 6-carbon molecule, and you get 2 pyruvic acid or pyruvate molecules produced. These are 3-carbon molecules. Sir, do we have to know this equation? No. There's lots of things that you don't have to know. What you do have to know is the things that I emphasize, right? In fermentation, 2 ATP molecules are produced, and initially as you saw before, 2 NADHs are produced. However, the NADH is used up in the second part or second half of fermentation, either lactic acid fermentation or alcohol fermentation. They're used up. So overall net production of energy compounds is strictly per glucose that originally is oxidized. It's 2 ATPs and no NADHs are produced in fermentation. That's a huge difference from the 36 or 38 ATPs total that is produced during aerobic respiration. Remember, we talked about 36 in eukaryotes and 38 in prokaryotes, 30 per ATPs produced per glucose. Here you end up getting 2 ATPs. So it's a big reduction from aerobic respiration. Remember, I told you that some organisms can carry out lactic acid fermentation, so they take the pyruvate and they further metabolize it down to lactic acid, or they metabolize it. It depends on the organism what kind of enzymes it carries, what metabolic capabilities it has, or they produce alcohol fermentation. Pyruvic acid therefore undergoes fermentation, and the end products can include lactic acid or ethanol. There are other possibilities besides lactic acid and ethanol, but we're not going to talk about those. So alcohol fermentation will produce ethyl alcohol and carbon dioxide. That's why in the fermentation of in production of wine or beer, you can get CO2 produced and you get ethyl alcohol. Lactic acid fermentation, you get lactic acid production, and some examples of organisms that can carry out lactic acid, bacterial organisms, bacterial genera that can carry out lactic acid fermentation includes Streptococcus organisms and Lactobacillus. End result is yogurt or sauerkraut or pickles or cheese. All of them. All of these are the result of lactic acid fermentation. I don't know why I don't have cheese written in here. I should. Cheese. Let's put it in like this. Cheese, please. All kinds of things. In many different cultures, there are many different products of lactic acid that are commonly used, that are commonly eaten. So lactic acid fermentation or alcohol fermentation from pyruvate. Now, when we look at metabolism overall, there's a wide diversity among different organisms, and they can all be divided into 4 types of nutritional forms or nutritional types of organisms based on their metabolism, where they get their energy from and where they get their carbon from, whether they're consuming or assimilating, bringing in carbon from other organic compounds or using CO2 gas from the air. And for energy, whether they're using light or chemical compounds like organic compounds for the energy source. So the 4 nutritional types are photoautotrophs or photoheterotrophs, and chemoautotrophs or chemoheterotrophs, where the first term, either photo, I'm gonna change colors here, where the first term, photo, refers to the energy source. The other choice is chemo. So that's for the energy source. Okay, for the first term there, whether they're using light or chemical compounds, like organic chemicals, organic molecules. That's for energy. And the second part of it is the carbon source, whether it's auto or hetero. Okay, auto refers to they do it themselves. They take directly carbon dioxide from the surrounding atmosphere and take the carbon out of that to build up organic molecules, chemical compounds, or whether they use organic compounds that come from other organisms, often dead organisms, to use as a source of carbon. And if they use CO2, then it's auto. And if they use other organic molecules, then it's hetero. So end result is we have photoautotrophs if they use light for energy and CO2 for carbon. Photoheterotroph if they use light for energy and organic compounds for carbon source. Chemoautotroph uses chemical organic compounds for energy and CO2, CO2 for carbon source. And chemoheterotrophs, and we are examples of chemoheterotrophs actually, use chemical organic compounds for energy. We break them down, and organic compounds for carbon source. We eat organic molecules, and many bacteria do also. So you have those 4 types, and that's shown here. All organisms. In green, in pink, it's the energy source, and in green, we're dividing up the carbon source. So actually, I shouldn't have said in pink because we're looking at both energy source and carbon source. First, we divide it up by energy source, where photo is for light energy and chemo is for chemical energy. And if you just look at those photoautotrophs, the phototrophs themselves, they can be further subdivided into photoheterotrophs, using light for energy and organic compounds for the carbon source, and photoautotrophs using light for energy and CO2 for the carbon source. I wouldn't worry about the examples shown here. And if they use chemical organic compounds for their energy source, these are chemotrophs, and they can be divided into 2 on the basis of the carbon source. Chemoheterotrophs that use organic compounds for the carbon, or chemoautotrophs that use CO2 for their carbon source. So chemoautotroph uses chemical for their energy and CO2 for the carbon. Chemoheterotroph uses chemical compounds for their carbon source and organic compounds for their energy. Chemical compounds for the energy source and organic compounds for the carbon source. Those 4 types: chemoheterotroph, chemoautotroph, photoheterotroph, and photoautotroph. Okay, so that's it. It's very brief for Lecture 5, Part 2. And it's up until this point that lectures 4 and 5 go together. 4 was an introduction to 5. Okay.